Our today's topic is detoxification, disposal of ammonia. In this topic, we will discuss how ammonia is trapped from the cells, then it is transported into the liver where it is detoxified and then excreted into the urine. So first step is the trapping of ammonia, which is the first line of defense. Second step is transportation of this ammonia to the liver, and the final step is the final disposal, that where in the liver it is converted into a less toxic substance, that is urea. Then it is disposed of in blood and then it reaches the kidneys and where it is excreted through urine. Now starting from the first step, ammonia is produced by almost every cell including neurons, and even minute quantities of this ammonia is very toxic to the cell, especially brain, as it causes cerebral edema. Therefore, it should be detoxified immediately. The intracellular ammonia is immediately trapped by glutamate to form glutamine, especially in the brain cells. The enzyme is glutamine synthetase. From there, the glutamine is then transported to the liver where the reaction is reversed by the enzyme glutaminase. The ammonia that is generated is immediately detoxified by converting this ammonia into urea. This we will study in urea cycle. Similarly, aspartic acid or aspartate may undergo similar reaction to form asparagine. Now, how ammonia is transported? The cells of almost all tissues produce glutamate, but there is an enzyme that is, I told you in the earlier lecture, glutamate dehydrogenase. This enzyme is only present in liver. Glutamate, it acts a link between the ammonia and the amino acid, and it is around 10 times more than all other amino acids in our body. The final deamination and production of ammonia will take place in the liver because there is this enzyme, the glutamate dehydrogenase, is only available in the liver. So final deamination will take place in the liver. Glutamine is the transport form of ammonia from brain and intestine to the liver. While the muscles use a different transport system. They use alanine to transport ammonia to the liver. These reactions we will study in detail. So please remember that from muscles, ammonia is transported through alanine, but every other cell uses glutamine as a transport form of ammonia. Why this we will study in this lecture. I'll explain it to you why. Now, final disposal of ammonia. The ammonia from all over the body, when it reaches the liver, it is detoxified, converted into urea by the liver cells, and then it is excreted through kidneys into the urine. Urea is the end product of metabolism, protein metabolism. If you are asked a question, what is urea? You will answer urea is the end product of protein metabolism. Although ammonia is toxic and has to be immediately detoxified, but in the kidney cells, ammonia is generated on purpose from glutamine with the help of glutaminase. Why? Because this is for buffering of the acid, and kidney helps to maintain acid-base balance by the formation of ammonium ions. Now, before we go into the detail of the lecture, there are some important terms that we must remember. Mammals, including human beings, excrete amino nitrogen mainly as urea. So they are referred to as ureotelic. Fishes excrete ammonia as such, so they are called ammonotelic. And birds and reptiles, they excrete ammonia as uric acid, so they are called uricotelic. Now, before we start the lecture, we have some important questions to remember. These questions will be answered in this lecture step by step, but you must remember these questions. Question number one is, what is urea and where it is synthesized? What happens to the pH when liver dumps urea into the blood? What if ammonia is directly dumped into the blood? Urea versus urate. What is urea? What is urate? Students get confused. From where the two nitrogens of urea come from? For this, first we have to remember or at least understand the structure of urea. This structure of urea is not asked in the exams, but for the understanding of this lecture and this urea formation, you must understand the structure of the urea. There are two amino groups that are attached to a carbonyl group, and this forms the urea. There are two amino groups. These are attached to a carbonyl group, and this whole molecule is urea. Now the second question, where it is synthesized in the liver, only in the liver cells. Now third question is, what happens to the pH when liver dumps urea into the blood? The pH will not change because these are amide nitrogens, and you know amide nitrogens are never charged, so there will be no change in pH. Next question is, what if ammonia is directly dumped into the blood? Now if ammonia is dumped into the blood, it will take hydrogen ion and form ammonium ion. OH minus will be left, which will raise the pH. So body incorporates ammonia into urea, then it gets into the blood and the pH is not changed. Now urea is a beautiful compound made by nature. It is sufficiently non-polar so that it goes right through the membrane and doesn't need a transporter, and it is sufficiently polar enough so that it can get absorbed into the blood, and it needs no carrier like albumin or anything, and it is also non-toxic. Now urea versus urate. Students can get confused between urea and urate. Now urea is the end product of nitrogen metabolism, that is protein metabolism, while urate, also known as uric acid, is the end product of purine metabolism. So remember this, don't confuse it with each other. Urea is the end product of protein metabolism, and uric acid or urate is the end product of purine metabolism. I will first start with urea and ammonia. Where does that urea and ammonia come from in the urine? Urea and ammonia, they come from kidney into the urine. Urea is the major product that constitutes the 86% of the nitrogen that is disposed of from our body, and ammonia, that is only 3%. The remaining percentage of the nitrogen is excreted through other molecules like creatinine and other molecules. So nitrogen is excreted, but mainly it is excreted as urea. Now we will start with the ammonia. From where does this ammonia come from that the kidney excretes into the urine? This ammonia comes from most of the tissues. In most tissues, there are a variety of deamination reactions going on. These deamination reactions are not only from the amino acids, but there are different reactions that are going on in the body tissues. These reactions, they form ammonia. This ammonia is put onto glutamate. Glutamate, as you know, glutamic acid is a dicarboxylic acid. It has two carboxylic group. One is on the alpha carbon. The ammonia is put onto the other carboxylic group to form an amide bond. This amide bond is formed and this glutamine is formed. The enzyme involved in this is glutamine synthetase. Now you should remember the difference between synthase and synthetase. It can be asked in Viva, what is the difference between synthetase and synthase? Difference is synthetase involves ATP while synthase does not need ATP. Now this glutamine, it is not charged because it has an amide bond. This glutamine is then dumped into the blood, and from the blood it goes to the kidney. Now in the kidney the reverse reactions take place. The glutamine is again converted into the glutamate by the enzyme glutaminase. In this way the ammonia is again released. This ammonia, the ammonia formed will pick up a proton and will form an ammonium ion. This ammonium ion is then excreted into the urine by the kidney. Now this is from where the ammonia comes from in the urine. That is from deamination reactions through glutamine. Now this glutamine that is formed, it goes into the blood, and apart from going into the kidneys, it also goes to another organ, and this organ is the intestine. Now this glutamine enters the intestine. In the intestine, glutamine is also formed from proteolysis of dietary proteins, and this glutamine formed is again converted into glutamate by the enzyme glutaminase. The glutamate thus formed releases ammonia, and this ammonia is also synthesized by the intestinal bacteria present in the intestine, and now this ammonia that is formed is dumped into the portal blood. Now here the question arises, previously I said that ammonia is not dumped directly into the blood because it can change pH. Now why in the portal blood? Because in the portal blood, liver quickly picks this ammonia up and takes it inside the liver, so it is not that damaging to our body. So ammonia is immediately transferred to liver, and from there it goes into the urea cycle where it is converted into urea. Now a point to remember here is that glutamine is the major transport form of ammonia in the blood. Remember this point, that glutamine is the major transport form of ammonia in the blood. Now another point to remember here is that this ammonia that comes from the intestine is the first nitrogen, one of the nitrogen that forms urea. We previously studied that urea has two amino groups, so one of the nitrogen in that urea comes from this ammonia. Remember that point. We will discuss this urea cycle later in the next lecture, but first we have to go to another organ, that is muscle. Now what happens in muscle? Now this usually happens when muscles are broken down under the condition of starvation. In starvation, our body needs glucose, and this glucose usually comes from the breakdown of proteins in the muscles during the conditions of starvation. Now, in the muscle, there is a family of aminotransferases. These aminotransferases, every aminotransferase is different from for every amino acid. If there are 20 amino acids, there are 20 different kinds of aminotransferases. All these aminotransferases will require pyridoxal phosphate that comes from vitamin B6, a coenzyme to remove that amino group from the amino acids. So this transaminase acts on amino acids to form alpha keto acids. Now this star is representing the nitrogen that will be transferred from one amino acid to another amino acid. So please do remember this. When this transaminase removes amino group from the amino acid, it will form an alpha keto acid, and this amino group is transferred onto alpha ketoglutarate and glutamate is formed. Now this amino group is transferred onto alpha ketoglutarate and this forms glutamate. Now this amino group is transferred onto glutamate. Now here another enzyme comes into play, that is called alanine aminotransferase. What it does, it transfers this amino group of glutamate onto the pyruvate and uses pyridoxal phosphate as coenzyme to form alanine. Now this amino group is transferred onto pyruvate and pyruvate is then converted into alanine. So in this way, this amino group, when transferred onto pyruvate, it forms alanine. Now this alanine is transported out of the blood, out of the muscle into the blood, and then it can be transported into the liver. It is the major source of nitrogen and carbon for gluconeogenesis and other reactions taking place in the body. Now this alanine does not change the pH of the blood because it is a neutral amino acid. Now the alanine formed is transferred into the liver. This alanine brought in from the liver is the source of nitrogen and source of carbon. Nitrogen is disposed of as urea and the carbon is used for source of fuel or energy purposes. Now this alanine is carrying the amino group. Now what happens to the alanine? In the liver, this alanine is again acted upon by alanine aminotransferase. What it does, it removes the amino group from alanine to form pyruvate and puts this amino group on alpha ketoglutarate to form glutamate. This enzyme also uses pyridoxal phosphate as a coenzyme. This pyruvate goes into gluconeogenesis. Now see that this amino group is now transferred onto glutamate. That is the nitrogen that alanine carried from the muscle is now transferred onto glutamate. Now here a question can be asked that why the body goes through this process of forming alanine from glutamate and then again this alanine is again converted into the liver into glutamate. Why not body straightforward sends glutamate into the blood and to the liver? Answer to that question is that glutamate will change the pH of the blood. It is an acid, glutamic acid. It will change the pH of the blood. So it is first converted into alanine. Alanine is neutral, it does not change the blood pH, and then when it reaches the liver, then it is again converted into glutamate. Now this nitrogen, it can also come from other amino acid catabolism that is occurring in the liver by aminotransferases, and this is put onto alpha ketoglutarate to form is glutamate. The amino acids are converted into alpha keto acids, and the nitrogen is put onto alpha ketoglutarate, which is then transformed into glutamate. And this reaction again uses the coenzyme paradoxal phosphate. Remember this, whenever there is an aminotransferase acting on an amino acid, the coenzyme will always be paradoxal phosphate. Now this glutamate formed can be acted upon by two enzymes. The first enzyme is glutamate dehydrogenase. What it does, it removes the amino group from the glutamate using NAD as a coenzyme and converts it into alpha ketoglutarate. The amino group is removed as ammonia, and this is the first nitrogen that forms the urea, one of the nitrogen that forms the urea. This ammonia is one of those nitrogens that form the urea. So this ammonia will enter into urea cycle, and it is converted into urea, and then it can be excreted into the urine. Now the second enzyme that can act on this glutamate is aspartate transaminase. What it does, it transfers the amino group of glutamate onto oxaloacetate to form alpha ketoglutarate, and this amino group, when transferred to oxaloacetate, it forms aspartate. Now this amino group, when transferred to oxaloacetate and it forms aspartate, this aspartate is the second nitrogen that forms urea. So there are two amino groups in urea. The nitrogen of one amino groups come from the ammonia, and the nitrogen of the second amino group comes from aspartate. Now ammonia has two sources. Now to summarize the last question, from where does the two amino groups or nitrogens comes from in the urea? Answer is the first nitrogen comes from aspartate and the second nitrogen comes from ammonia. Now this ammonia itself comes from two sources. Number one is it comes from glutamate, and the second source, it comes from the portal blood that comes from glutamine from the intestines. So to summarize, first nitrogen comes from aspartate and second comes from ammonia. Now this aspartate that is formed, it enters the urea cycle. Now you have to remember two enzymes. Number one is alanine amino transaminase that converts alanine into glutamate by transferring its amino group onto alpha ketoglutarate to form glutamate. In this way, pyruvate is formed, which enters gluconeogenesis. And the second enzyme is aspartate transaminase that converts glutamate into aspartate. So with this, we finish our lecture, and in the next lecture, we will study the urea cycle.